Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to your channel The Anatomy Canvas. Today we are going to start with the embryology of the gastrointestinal system, and in today's class we will do the foregut. So learning objective for the today's class are division of the gut tube, the molecular regulation of the gut tube development, and to discuss the development of the mesentries and describe the development of the esophagus. So the end of the lecture you will understand all these terminologies and all these topics. So gut development, the primordial gut forms during the 4th week of development as the head fold and the caudal eminence and the lateral body folds incorporate part of the dorsal part of the yolk sac into the body cavity. So primitive gut is divided by head and tail fold into 3 main compartments. The foregut that extends from the buccopharyngeal membrane. Here is the buccopharyngeal membrane, and foregut starts from here and to its continuation into the central midgut region via cranial intestinal portal, also called the anterior intestinal portal. The cranial end of the foregut, the embryonic pharynx, is intimately associated with head and neck development. So this area is the embryonic pharynx, and it is associated with the development of the head and neck region, and it extends from oropharyngeal membrane to the respiratory reticulum. The midgut extends from the intestinal portal from the anterior intestinal or cranial intestinal portal to the posterior or caudal intestinal portal. And the hindgut extends from the caudal intestinal or posterior intestinal portal till the cloacal membrane. So these are the 3 different parts of the gut tube which are incorporated into body cavity because of the folding of the head, folding of the caudal eminence, and lateral folds of the embryonic body. So this wide connection like a purse strings to suture is incorporated into the body of the embryo. The differences between the portion of the gut tube develop as a result of interaction between 3 embryonic tissue layers which give rise to the gut tube, namely the endodermal lining, the epithelium. So this red color shows the endodermal lining of the endodermal inner epithelium, then the thick splanchnic layer which is derived from the mesoderm in the area, and then the outer layer proliferating splanchnopleurotic coelomic epithelium. So these 3 areas, these 3 different type of the tissues interact with each other, and as a result of these interaction different parts of the gut tube will be formed. So in this diagram you can see this area is the pharynx, and this is the bulge of the pericardium cavity, and behind this bulge you can also see the respiratory diverticulum as well. This area are the pericardio-peritoneal canals, and green color areas show the septum transversum which is developing over here. Then this is the wide connection where the midgut is connected in a wide to the yolk sac, and then this area is the hindgut where you can see the cloacal membrane, also allantois is present over here. So cells of the origin of the gut tube also important from your papers MCQ point of view. So there are different origin for the gut tube. The epithelium is derived from the endodermal lining epithelium of the gut tube. The lamina propria, the muscularis mucosae, the connective tissue of the submucosa, muscularis externa, and the external connective tissue all are derived from the splanchnopleurotic mesenchyme. And outer to this, the peritoneal epithelium, that is the serosal layer, is derived from the splanchnopleurotic coelomic epithelium. So these 3 are the sources from which the gut tube developed, that is the endodermal epithelium, the splanchnopleurotic mesenchyme, and splanchnopleurotic coelomic epithelium. So please remember students, these all are your MCQs as well. This is a diagram to show that this is the area of the pharynx, and here is the buccopharyngeal or oropharyngeal membrane, and this part is called the stomodium. Then this is the area of developing thyroid and lung bud over here, and developing area of the heart, and developing area of the liver. And this area is the midgut, and here is the hindgut where you can also see the cloacal plate. This is the tail fold and the allantois twice present in the body stalk, and this is the posterior neuropore, and here is the anterior neuropore of the neural tube. So first of all, there is a different concentration gradient of the retinoic acid along the whole gut tube, to the minimal level in the foregut region to the maximum level at the hindgut region. So retinoic acid gradient actually specifies the different areas. Then you should remember that there throughout the gut tube there is an expression of the sonic hedgehog protein throughout the endodermal lining of the gut tube, and there is interaction between the sonic hedgehog protein and its surrounding mesenchyme. As a result of this, in epithelial and mesenchymal interaction, different genes are initiated. So first look at this diagram. This purple color show SOX2 gene, which is responsible for development of the esophagus and the stomach. Then this blue color area show PDX1 gene, also called the pancreatic and duodenal homeobox genes. So expression of these genes results in formation of the pancreas and the duodenum. Then the two genes, the CDXC and CDXA, also called the caudal genes. So these two genes are responsible for development of CDXC genes responsible for development of the small intestine, and CDXA gene is responsible for development of the large intestine and the rectum. So please remember the role of the different genes over here. So first was the CSOX2 gene, then was the PDX, also called the pancreas and duodenum homeobox gene. Then is the CDX gene, also called the caudal genes. These all are the different types of the homeobox genes. And the last is the HOX gene, which is homeobox gene in green color. This shows development of the liver. So different genes which are involved over here. This is the molecular basis of the GIT formation, which will lead to expression of the epithelial and mesenchymal interaction, lead to expression of these different genes, and their gradient varies, which results in specification of different parts of the gut tube, which will form different areas like small intestine, cecum, large intestine, cloaca, etc. So various stages of embryonic development. This diagram, a beautiful diagram in which you can see the folding of the embryo in different direction. This is the somatic mesoderm and this is the embryonic, and this whole is the yolk sac. So because of the folding of the embryo, this yolk sac is incorporated into the body cavity, and initial there is a connection between the gut tube and yolk sac, which will later be diminished, and this yolk sac initially in a wide connection with the dorsal body wall. Later it is suspended with the dorsal body wall by the dorsal mesentry. So because of the folding of the embryo, the part of the yolk sac is incorporated into body cavity, forming the gut tube. So formation of the primitive gut, that is because of the cephalocaudal folding and lateral folding of embryo, leads to formation of the blind ended tube from the cephalic end to the caudal portion of the embryo. At the cephalic end there is the oropharyngeal membrane, and at caudal end there is the cloacal membrane. So pharyngeal gut, that is pharynx, extends from the buccopharyngeal membrane to the lung diverticulum. The foregut extends to the liver diverticulum. The midgut extends to the junction of the right 2/3 and left 1/3 of the large intestine, and the hindgut extends to the cloacal membrane. So this diagram shows different areas of the gut tube at day 26 of the embryo, which develop into different parts. This green color area show the pharyngeal gut. Here is the stomodium, and here is the membrane called the oropharyngeal membrane. This is the respiratory diverticulum. This is the hepatic diverticulum, and this is the foregut. So foregut basically pharyngeal part, and the part which forms the esophagus till the opening of the diverticulum of the liver. Then the midgut shown in blue color, and then the hindgut, and this area is the cloaca, this is the allantois, and this is the opening of the connection of the gut tube with the yolk sac. So this is a lateral view of developing 26-day-old embryo showing different parts of the primitive gut. The foregut begins caudal to the respiratory diverticulum as you see here and extends up to the liver diverticulum. So what are actually the mesentries? The mesentries are double-layered fold of peritoneum that connect the gut to the ventral and dorsal body wall. This is very important definition. You should all know what is mesentry. So double-layered fold of peritoneum that connects gut to the ventral as well as dorsal body wall. So the organs which are present inside the peritoneum, they are attached to anterior as well as posterior body wall by the peritoneum are called intraperitoneal organs, and the organs which have only the peritoneum on their anterior aspects and at their dorsal aspect, they are in direct connection to the body wall. They are called the retroperitoneal organs like the pancreas. The peritoneal ligaments are the double layer of the peritoneum which either attach two organs to each other or attach one organ to the body wall. So next important definition is the dorsal mesentry. It extends from lower end of the esophagus to the cloacal region. This is important to understand. From lower part of esophagus to the cloacal region, and in stomach it forms the greater omentum, and in small intestines it forms the mesentry proper. So dear student, must remember these all are your MCQs. So stomach, in the area of the stomach, the dorsal mesentry is called greater omentum, and in area of the small intestine the dorsal mesentry is called mesentry proper. These are your MCQs, please remember. This is the diagram to show the position of the dorsal as well as the ventral mesentry. So this brown color area show all of the dorsal mesentry from the area of the lower part of the esophagus till the cloaca. The all of the GI tract is attached to the dorsal body wall. In the area of the stomach it is called greater omentum, and in the area of the small intestine till cloaca it is called the mesentry proper. In the region of the large intestine, it is called dorsal mesocolon. Also remember that the special artery, the celiac artery, is the artery of the foregut which run in the mesentry. This is the stomach, and this whole area is the ventral mesentry. Because of the developing liver into the septum transversum, this ventral mesentry is divided into two parts. The part which lies between the lower part of esophagus, stomach and upper part of duodenum is called the lesser omentum, and part of the ventral mesentry which lies between the liver and the anterior abdominal wall is called the falciform ligament. So again these two are your MCQs as well. So this is area is the bare area of the liver where it is in direct contact with the diaphragm. Here you can see the midgut and the hindgut. So what is the ventral mesentry? Extending the region of the terminal part of the esophagus and upper part of the duodenum. It is derived from the septum transversum. Again your MCQ. Growth of liver in the septum transversum divided it into two. The lesser omentum extends from lower part of esophagus, stomach and upper part of duodenum to the liver, and the falciform ligament extends between liver to anterior abdominal wall. So please remember that these are your MCQ both in gross anatomy as well as in embryology. So now we move on to the development of the foregut, and in development we will do the esophagus first. So in a 4-week-old embryo, the lung bud appear on ventral part of the foregut. Here in this diagram you can see this is the ventral aspect of the foregut where the lung bud is appearing as a respiratory diverticulum. Later on the tracheoesophageal ridges appear, and when these ridges meet in the midline they form the tracheoesophageal septum, and this tracheoesophageal septum then divide the foregut into ventral part, that is respiratory diverticulum, later on forming the trachea and the lung buds, and the dorsal part which will form the esophagus. So please remember Muscular coat of the esophagus is formed by the splanchnic mesoderm, and it is striated in upper two-thirds, and it is derived from fourth and sixth pharyngeal arches, and lower third part formed by the smooth muscle and derived from the splanchnic mesoderm. So dear student, please remember, these are your MCQs. Their origin, that is splanchnic, they are striated in upper third and derived from the fourth and sixth arches, and the origin in the lower third, that they are smooth in the lower third and they are derived from the splanchnic mesoderm of the area. So splitting of the foregut into esophagus and trachea, these are the tracheoesophageal ridges or septum, which are dividing the anterior and the dorsal parts into the respiratory diverticulum and the foregut. This is forming trachea and lung bud, and this is forming the esophagus. So these are the different anomalies, the tracheoesophageal fistula. In these different slides, you can see there is a deviation in the tracheoesophageal septum, as a result of which either the proximal part of the esophagus ends in a blind ending tube and lower end or distal end has a connection with the trachea, or in this case both the tubes ends blindly, or in this case they have a fistula with the communication with the trachea. In this case the upper part has a connection with the trachea and the lower part ends in as a blind ending tube, and in this diagram you can see both the upper and the lower part enter separately into the trachea. So these are the different tracheoesophageal fistulas because of the incomplete separation and or atresia of the trachea and esophagus. Defects likely in mesoderm and usually associated with other defects involving the cardiovascular information like vectoral, etc. So what is the vetor? Vetor is vertebral anomalies, the anal atresias, the tracheoesophageal fistulas, esophageal atresias, and the renal atresias. And vectoral is vetor plus cardiac defects and limb defects. Again, please remember, these are your MCQs as well. So here, dear students, I complete my today's class. The references are the Langman's Medical Embryology, The Developing Human by Keith L. Moore, and The Gray's Anatomy by the Susan Stranding, the 40th edition. Thank you very much for your attention for the today's class, and we will meet inshallah in the next class. Till then, Allah Hafiz.